So hello from my side. So I'm coming from pediatric hospital, working as a bioinformatician with sequencing data. So I will give you a brief overview of the project that we are into it to improve the sequencing and rare disease diagnosis. So I'm in the laboratory for diagnosis of rare diseases. So since each, I will go about introduction, what's the idea and the background of all the research and development that we are doing. So since each rare disease is rare, but the number of the rare diseases is huge. So it's around 8,000 known rare diseases, and 80% of rare diseases is caused by mutation in DNA. And in Slovenia, actually, there is not so low number of the people living with rare diseases, so it's around 4 to 7%, which makes up to 120,000 of Slovenians having rare diseases. Time to diagnose it's 7 years in average. These are complex diseases, and 40% of patients are being misdiagnosed. So this is the reality that we are facing on pediatric hospital. And the treatment, over 90% of rare diseases have no approved treatment. So I will go a bit in the basics not with the formulas, but the basics. So we have a cell, we have a nucleus and the chromosomes in it, 23 chromosomes, which are composed from base pairs, so amino acid, adenine, thymine, guanine, and cytosine. In human genome, it's 3.2 billion base pairs, so these nucleotides. And the change in one can cause a complex disease. So what are we actually doing? It's just looking through these 3.2 billion base pairs and searching the one that is causing the phenotypes that it's reported by clinicians. So biology, we all know it's not like just DNA. We have several layers of it. So we need also to explain, to find the connection between this change and the phenotype. So the technologies that we are doing are from sequencing to RNA sequencing to structural variations, metabolomics, single-cell profiling, and to explain the phenomena that we see in clinics. So I will, the new technology that will be also part of a later talk, it's sequencing technology. When we pull DNA through the membrane, record the ionic current that is going through this membrane, and with the training models, we get then the base pairs that are composing this DNA. So for genomics and use genomics in clinics, I really like this quote. So the progress depends first on new techniques, then discoveries and ideas. So the advancement in technologies, in computation, in genomics, it's really important for us to find more, to put more patients to diagnosis. So this is the reality from our lab. So we have in the last days, in the last years, really hyperinflammation of sequencing due to the genomics technologies, and we need to actually follow this with all the computational power. So if we were facing in 2010, 2020 with few terabytes in few years, now we are facing with up to 30 terabytes in few days of this data in ionic current that we need to process as fast as possible to find out the right mutation that is causing the disease and to guide treatment. So this is behind the data. We have a processing pipelines, and we don't know what to do without GPUs, of course, because like calculating this on CPUs would be, it's useless, it takes months. And advancement with GPUs, it's of great advantage to us. We are getting to analyze the data faster and faster and faster. So we are really trying to improve all this data with the hardware that it's available up to date. So from the processing, we are part of European initiative. Since we are dealing with rare diseases, when we have just one patient or two patients in Slovenia, there are few patients in Europe and few patients in the world, we need to do a cross-border access to genomic data, to share the data, to share the phenotypes. And with genomic data, which are really sensitive, the security of this data, it's a crucial importance. So we should not place the data out of Slovenian territory by the law, and that's usual for all the European countries. So we get to how to share the data if we cannot share it by the law. So that's why we are using these all European initiatives. And starting with to see what's abnormal and pathologic in our case, we need to know what's the present in the healthy populations. So we have a project founded by ARIS, Slovenian Reference Genome Project, where the first goal is to sequence 500 healthy volunteers. And then we will go up to 2% of Slovene population. So this is part of a larger Genome of Europe project. And then to analyze, to share all this data, we are part of European Genomic Data Infrastructure project. And this is all under umbrella of 1+MG initiative. So European Genomic Data Infrastructure, here I have like just a brief overview how the technological part is structured. We have 5 different stages of sharing the data, parts of sharing the data. This is from data discovery, so from the user portals, data catalogs, then to log into the system, where you have authentication and authorization infrastructure, to search the right data that you want to inspect, to apply for this data, to have approved from some data committee that you can access to this data, and then to get this, the right data that you want. The project is going also to the federated computation, so to have data from multiple centers, not seeing it, train the model, and then pull the models together to improve all together without actually accessing the data. One use case of this genomic data infrastructure is Genome of Europe, where the goal is to sequence 100,000 genomes from healthy populations around, across 27 countries. And as I mentioned, we are part also of Slovenian Reference Genome Project, where right now, currently, we are sequencing first 500 healthy volunteers. And we will include this in Genome of Europe project. To describe you better how the Slovenian node of data infrastructure for this genomic data looks like, this is led by a team of Professor Oysteršek from Maribor. So we have a Slovenian GDI node that is connecting to European GDI infrastructure, and local different institutional GDI nodes. And this then we can access genomic data archive, compute things through the Slink network, and having Slovenian open science infrastructure. So from our perspective, to be in more detail, this is like we have sequencing on our institute and on the Institute of Medical Faculty. We are sequencing the data, we do the data analysis and all QCs that needs to be evaluated, that the data are quality enough. And then we have genomic files, final genomic files with phenotyping metadata, which are sex, country of origin, and age. The data are de-identified, so they are anonymized, they are encrypted, and then we have secure upload to the national node. In the national node serve as a secure environment for storing and sharing this data. And there is then recalculated to have some catalog of healthy Slovenian genomic variants. So in more detail, for this national node, it has its own local user portal, contained computational part, genomic data archive, and then it has a resource entitlement management system, so everything what it was built for these genomic systems. It is connected to European services, and it's connected to our local infrastructures, centers. So this is just a visualization of Slovenian Genomic Data Infrastructure portal. Right now we have only synthetic data in it because we are still developing it, but in the following months we'll starting to fill it the data from Slovenian Reference Genome Project in it. So that would be all from my side. I would like to thank you to all the things won't go without cooperations between University of Ljubljana with medical faculty, with University of Maribor, Laboratory for Heterogeneous Computational System, and all the co-workers from Pediatric Hospital. Thank you.
Thank you very much, Barbara Jenko Bizjan. Any questions for her, considering this very interesting topic? We are all looking forward to seeing what will be the final results of having this 500 genome sequenced and then analyzed. When do you plan to finish that, the first stage?
We are already sequencing it. I hardly estimate how, like in 1 year, I think we will have sequenced all 500.
500. Yes. And then you said 2% of the Slovenian population?
Yes, these are then another stages. Here we need to have involved all minority groups in Slovenia, all the other things to have. But yeah.
And that will last approximately?
This is just planned for next 10 years.
Next 10 years.
Apart just from DNA, we are planning to collect also the RNA sequencing to have a better eye-half of how is the situation in healthy population. And especially in Slovene, because we are working with Slovene patients, and then it's much easier to detect the variation that is causing the disease. Mm-hmm. That's where we come from.
Will there be anything like very uniquely about the Slovenian genomes that we are maybe different from the, I don't know, from the Austrian?
When we are looking to PCA plot, which is like the plot that it's for from genomic variation, Slovenia is somewhere a bit away from the others. I can tell this. I can put it in another presentation.
In which characteristics?
I don't know, because it's just like whole genome. Okay. Hardly to say some specialty.
Any questions there? From the this is the microphone. Just wait for the microphone to come to you.
Yeah, thank you for your talk. I'm wondering, like, of course, all of this data also by law has to be very tightly protected to also protect the integrity and everything of patients. But I was wondering, because the majority of these diseases don't have treatments, and like, how do you then, like, are there research groups, for instance, that are able to still mine these data anonymously to look for variants and like then connect it to the proteins and maybe like how they could be targeted for drug discovery? Are there ways to actually access this data, or is this like currently just for like still being stored and closed access?
Yeah, currently it's stored and closed because on this point of we didn't develop, all the system needs to be developed for our hospital I'm talking. But we are going in this direction. With all this project, the first thing is we need to do all ethical considerations, which is actually, we found it harder than a technical consideration to overcome. It's just crazy. And then when we get through all the ethical considerations, through all the documents needs to be written, the idea of all these projects and the long-term project is to have this data being more accessible in a very secure and authorized way for the development of treatments and for the better diagnosis. But for healthy volunteers, it's easier. You cannot really so easy to define like person from the genome. But from the rare disease patients, we all know the mutations that these few patients have. So if I will see a genome, I will know from who is coming. So here is the privacy of the data. It's here of crucial importance. So that's why the improvement, advancement in the clinics is going much slower than in the research. So, yeah.
Thank you.
Any other question? Otherwise, Barbara Jenko Bizjan, thank you very much for your talk, and a large round of applause for her.